Okay, today I'm going to teach you how to use EasyGel to check and see if there is E. coli in your water. Alright, before you do anything, you want to make sure you wash your hands with soap and water. Make sure you get a lot of your hands with a lot of soap, nice and bubbly, backside, between your fingers, everything. Make sure they're completely sterile. Now, a lot of times in a village, the next thing you would do after you turn off the water is you'd grab for the local towel. But the local towel probably has as much microorganisms in it as anything else in this entire area. So just let your hands, shake your hands dry, and then let them air dry before you start to work. Alright, so the first thing you need to do is get ready. So I'm going to open up a fresh bag of EasyGel Petri dishes. These Petri dishes are not just regular Petri dishes. You can't see it, but they're coated with a special chemical. Okay? So you need, for one test, you need one Petri dish. Okay, and the other thing that you're going to need is a solution. So again, this is EasyGel. Okay? And I'm going to take, for each test, I need one container. Now, we keep them in the freezer, and that keeps them fresh. So, what we need to do is put that rest back in, but I'm going to, because it's frozen, I need to make it liquid. So I'm going to just put it in a cup and then pour hot water over the top of it. Alright, and then we just have to wait a few minutes until it's dissolved. Okay. Alright, after it's been soaking in hot water for maybe 5 minutes, check it. Just kind of swish it back and forth, make sure you don't hear any sort of ice cubes in it. You can also feel it to make sure that it's rather warm. Alright, then after you do that, you need to prepare your sample. I have samples already, and they're in the refrigerator, so let me go ahead and just get it out. Okay. Alright, this is the water that I've already sampled in the previous video from the faucet. Okay, so what I need to do is get this water into a petri dish so the microorganisms can grow. So there's two different ways of getting the water out. One is a pipette method, okay, and I have a 3 milliliter pipette. I need to get 5 milliliters of sample water for drinking water into this container right here. So what I've done is I've already marked it with a Sharpie between the 3 milliliter line and the 2 milliliter line, and so it's 2.5. So if I use this method, what I would do is I would take the water out twice. Okay, the other method is a little bit easier because I happen to have a graduated cylinder, and then I could fill it up. So let me just start with that. Alright, so again, my hands are already washed and clean, and I'm going to carefully open this up, don't put it down on the ground, and I'm going to pour the water in. Now again, I'm going to assume that this graduated cylinder is dirty, so I'm going to fill it up once, twice, and the third time I'm going to count it good. Now it's actually very difficult to get right at 5 milliliters. So just for this video, I'm going to say I'm right at 5 milliliters. Okay, so what I would do then is I would open up the EasyGel container, and I would pour the water directly in. Okay, now let me show you now the second method. The second method is using this pipette. Now I've put this on a dirty surface, so that wasn't a good example. So you need to make sure that you put it on a clean surface. So for this video, let's just assume that I had laid it on a clean surface. So likewise, now what I need to do is I need to take off the lid, hold on to it, and I'm going to carefully stick my pipette in. First I'm going to squeeze about all the air out of it. And then I'm going to carefully let go of it once it's in the water and pull it up. Okay? Once it hits that black line that I've marked with my Sharpie, go ahead and pull it out of the water. There may be a little air bubble, and that's okay. All right. Now, I need to take this off while not squeezing any of the water out of the pipette. Okay. Okay. And that is 2.5 milliliters going in. Okay. Now the next time is the second. Okay. And cover it up. Okay. Now, because I'm done using this pipette, I can put it on a dirty surface, no problem. Put the lid on this, put the lid on this, and now carefully, I'm just going to turn this over a few times. And I'm going to do this so that it doesn't get air bubbles, but it mixes well enough. Okay. Then you directly go ahead and take the lid off. Carefully take this off. Make sure that you have the top, okay? See, the top is larger than the bottom. Okay, and slowly pour it in. Okay, now after you pour it in, you kind of shake it, make sure you have all of it out, but no, you're not making any bubbles. Now you're going to let it sit there for about 1 hour until it's solidified. You can see right now that it's liquid. I just poured it in, but in about an hour it's going to solidify because it mixes with the chemical in this plastic Petri dish. Okay, after you've poured the solution into the Petri dish, and while you're waiting for it to solidify, let's make sure that we write down important information about it. So this is going to be the name of the place, so that it would be the tap from Filter Factory. Nice and clear, so even in a few weeks you could remember. Then how much of the water sample did you put in? 5 milliliters. All right, and today's date, which today is 8/23/2013. Okay. Okay, after waiting about an hour or so, we look at it, it's pretty solidified. Okay, so then we're going to carry it, and in our factory we have an incubator. The incubator is set for 37 degrees. For this type of testing, you want the air temperature to be between 25 and 40 degrees centigrade or Celsius. Okay, so we have a professional incubator. The key here is that when you put it, you want to put it upside down, and that's why we had to wait for 1 hour before it solidified. Okay, you put it in the incubator, upside down, close it up, and now you need to wait 24 hours. Okay, so after pulling it out of the incubator, after 24 hours, we can see that that water really is contaminated. So try to get up as close as you can here. You're going to see there's just an awful lot of blue dots. Blue for this sampling is the E. coli. But I'm going to hold it up to the light here, okay, the outside light. And that's going to help me a little bit to sample. So you can see there's one here, one here, one here, one here, one here, one here. Just in a small area, I already have 6. Okay, but you need to go through slowly and count all of these blue dots. All right, so let's say that we counted 80 blue dots. That's 80 E. coli in 5 milliliters. The standard is 100 milliliters. So you'd have to multiply 80 times 20, which is 1,600 E. coli per 100 milliliters. The standard is not 1 per 100 milliliters. So this water is very polluted, and you definitely would not even want to wash your hands with it. Okay? All right, I want to explain in general how to interpret your results. You'll see here is an idealized petri dish, but you'll see that there's plenty of colors on different colonies, and these colors represent different types of microorganisms. The first color I want you to look at is the blue or purple color. These are E. coli. E. coli is an indicator organism that there's fecal contamination in the water. So if you have this type of E. coli, that means that there is fecal contamination in the water that you have tested. The other color, second color I want you to look at are the pinks or reds. These are Aeromonas. Aeromonas are associated with diarrhea, vomiting, abdominal pain. Basically, if you're seeing this in the water, you're going to have a very high prevalence of diarrhea, where with E. coli you may or may not have it. Then the third color I want you to take a look at are these green circles here, and this is associated with all sorts of Salmonella species. And so you may be familiar, Salmonella is another one that causes diarrhea quite a bit. And so again, this is a great concern. So E. coli is what we use to measure and report. But if you're seeing these other colors in there, then you have grave concern for people who drink this water that they're going to be having serious diarrhea. On top of that, you may also be seeing other colors like yellow or clear or white. Those are going to be other microorganisms, but this test cannot identify them clearly. All right, after you have just looked at all those microorganisms, you may not realize it, but it's really easy for those microorganisms to get on your hands. Each little dot represents 1 billion microorganisms. All of those microorganisms can make you sick. So the very last thing that you need to do, again, is get some good soap, turn on some clean water, and wash your hands before you leave the area. All right? Before you type on your computer the report how many microorganisms you found in the water, before you do anything else, wash your hands.